Hello student, good morning. Today I will teach you oogenesis. Oogenesis. The process of oogenesis occurs in the cells of the germinal epithelium of the ovary. Such cells are known as primordial germinal cells. The oogenesis is completed in the following three successive stages. Oogenesis completed in the following three successive stages. Number 1, multiplication phase, growth phase, maturation phase. First is, that is, we have to see multiplication phase. For oogenesis, some cells of germinal epithelium becomes large sized. For oogenesis, some cells of germinal epithelium becomes large sized and multiply mitotically to form the population of egg mother cells or oogonia. When mitosis stop, the oogonia are called oocytes. The oocyte enter the prophase of meiosis. First and now are called primary oocyte. Before the completion of first meiosis, the primary oocytes pass through a phase of growth and differentiation. Note that oocyte nucleus is tetraploid since it has already passed through the S stage. S means synthesis stage of interphase to double the normal diploid DNA content. Second, growth phase. First is multiplication phase and second is, that is, the growth phase. In this phase, the growth phase of the oogenesis is comparatively longer than the growth phase. The growth phase of the oogenesis is comparatively longer than the multiplication phase of the spermatogenesis. Growth phase of the spermatogenesis means oogenesis process is longer than the growth of spermatogenesis. In the growth phase, the size of the primary oocyte increase enormously. For instance, the primary oocyte of the frog in the beginning has the diameter about 50 micrometer, but after the growth phase and diameter of the mature egg reaches about 1000 micrometer to 2000 micrometer. In the primary oocyte, large amount of fats and protein becomes accumulated in the form of yolk, and due to its heavy weight or gravity, it is usually concentrated towards the lower portion of the egg, forming the vegetal pole. The portion of the cytoplasm containing the egg nucleus remains often separated from the yolk and occurs to towards the upper side of the egg, forming the animal pole. During growth phase, following changes occurs in the nucleus and cytoplasm. Nucleus changes. In growth phase, nuclear changes. Following changes have been observed. Number 1, growth of nuclear material. The nucleus of primary oocytes increase in size due to the production of large amount of nucleoplasm. Consequently, the nucleus of the advanced oocyte appears to be swollen and is usually called as germinal vesicle. The chromosomes increase in length, but the amount of DNA is chromosomes does not increase in proportion to the enlargement of the nucleus. Second number, formation of lambrous chromosomes. In the amphibian eggs and large yolky eggs of fishes, reptiles and birds, the chromosomes acquired a very characteristic appearance in the diplodine stage of one first meiotic prophase. The diplodine stage lasts for 3 years in amphibia. Many loops are thrown out transversely from the main axis of the chromosomes up to 10,000 per chromosome set in salamander. The loop represents the loci of gene activity. Messenger RNA is synthesized at these regions. The messenger RNA later controls and guides the protein synthesis in the cytoplasm of oocyte during the growth phase. These loops are withdrawn after the diplodine stage. Third number, in chromosomes, all the messenger RNA molecules are not utilized for protein synthesis during the growth phase. Means all the messenger RNA not utilized for the protein synthesis. Some messenger RNA synthesis the protein, but not all messenger RNA. During growth phase, some of they are inactively, some are active and some are inactive. By the wrapping, when the proteins and stored these are called as informosemes. Nuclear changes. The nucleolus of primary oocyte is involved in biogenesis of ribosomes and enlarged considerably in amphibian oocytes. Instead of one, a large number of nucleoli, example about 3000 per xenopus oocyte, are formed on the periphery of germinal vesicle. The increase in the number of nuclei is due to increase in the number of genes coding for ribosomal RNA in nuclear organizer. The multiplication of genes without mitosis is called as selective gene amplification. Growth of cytoplasmic materials. The cytoplasm of primary oocyte increase undergoes following changes. Mitochondria, which is the changes of mitochondria in growth phase. Number of mitochondria increases considerably. In amphibians and birds, mitochondria aggregate in the form of a large mitochondrial clouds. Roman of 1960 and Wartburg in 1962. Belinsky and Davis in 1963 due to the increase in their number of oxygen consumption increase during the growth phase of oocyte. Mitochondria contains oxidative enzymes. Ribosomes. Oocytes contains numerous ribosomes in their cytoplasm for the increased protein synthesis during the oogenesis. Endoplasmic reticulum. Most oocyte contains the smooth endoplasmic reticulum. Smooth endoplasmic reticulum. SER means smooth endoplasmic reticulum of small vesicles which are dispersed evenly through the cytoplasm. Rough endoplasmic reticulum RER is often not found in your young or mature primary oocyte, since the protein are being synthesized for storage rather than for secretion. The storage proteins are normally synthesized. Storage protein are normally synthesized. Some proteins also are storage and that also synthesize on the free ribosomes. The growing oocytes also contains annulate lamellae in their cytoplasm. These are membranous structure which appears as the stalk of cisternae. Golgi apparatus. These are found around the centrosome of younger oocytes, but in mature oocyte the disappears completely. The Golgi apparatus produce cortical granules. It produces cortical granules. Cortical granules means what? These are found in the oocytes of frog, fishes and some mammals, as rabbit and man also. They form a layer close to the plasma membrane in mature oocytes. They are spherical diameter, 2 mm in frogs, remains surrounded by the simple membrane and contain the acid mucopolysaccharides. Vitellogenesis. Yolk appears in the oocyte in the second period of their growth, called the vitellogenesis period. Yolk is the most usual form of food storage in the egg in the amphibians. Some scientists, that is the word in 1962 and Belinsky and Davis in 1963 and the fishes Yamamoto and Uta 1967, the vitellogenesis or synthesis of different yolk components takes place inside the modified mitochondria in other vertebrates. The yolk is not synthesized in the oocyte at all, but is produced in the liver of the body of the female. It is then transported in a soluble form via the blood streams and the follicle cell to the oocyte, when it is finally deposited in the form of yolk platelets or yolk granules are protein, phospholipids and fats in the different combination. Function of yolk. Yolk is the most usual form of the food storage in the egg. It influences the differentiation of ooplasm and the patterns of the cleavage. The size of the egg is determined by the amount of yolk present in it. Yolk exercise an important influence on the morphogenetic movements of the blastomeres during the gastrulation. Okay, thank you.